I'm delighted to have Ruth Baker from Oxford talk today about a topic that I find endlessly fascinating and confusing. I look forward to Ruth just putting some sort of order and clarity into these terms, identifiability and inference in cell biology. So Ruth did her undergrad degree and doctoral studies in Oxford, worked closely with Philip Maney, spent then some time here in Melbourne before returning to Oxford, where she's now running a group that combines mathematical modeling at multi-scale levels, so going all the way up to tissues and statistical inference. And I look forward to your talk. And Ruth, the floor is yours.
Thank you, Michael, for the introduction and for the invitation to speak. I should have said that I swapped out part of this talk, so it's probably a lot less about identifiability than it has been of late, but nonetheless, hopefully it's of interest. So I thought just to start with, I'd sort of give a bit of an introduction to me and the group here in Oxford in terms of our research focus and the way in which we try and do things. So we've had a long fascination really with the mechanisms that drive collective cell motility, so over very long distances, and then how these sort of mechanisms combine with cell adhesion, cell proliferation and death to establish really complicated biological processes. And much of our focus has been on developmental biology and developmental processes, but really with a view to thinking about, needing to understand sort of processes that develop tissues and organs so that we can understand kind of how to tackle sort of various diseases, so pathologies that occur when these processes become misregulated, or things like wound healing and regenerative medicine, to how to grow tissues de novo. And our goal increasingly, I would say over the past few years, has been to look at multiple types of quantitative data and to try and interrogate those data using validated and predictive models to provide new insights. And that's always really the goal. So I know I'm preaching to the converted, but just as a sort of to highlight, we especially, well, in applied projects, we really try to take a sort of tight collaborative approach where we're working with experimental groups who are looking at both wild type and perturbed scenarios, and we're developing mathematical models and means to simulate those models, and then sort of working together to analyze the data and to test models on hypotheses. And we're always aiming for this sort of predict, test, refine, predict cycle. So for example, we might have experimental data which has led to the development of some hypotheses for a given mechanism. And our aim is to encode those hypotheses in a mathematical model where we can essentially sort of test them against the observations that we have in the data. And I always say that we always learn the most essentially when the predictions or the testing of our mathematical model throws up some inconsistencies, because then it tells us that something essentially about the workings of the biological system that we're thinking about that we don't understand. And we can then use the modeling framework to go away to refine our hypotheses and then essentially make experimentally testable predictions. And for me, it's really this cycle that I think is so important within sort of quantitative biology, if you like. So what I'm going to do today is to try and talk about some different examples where we've sort of tried to attempt this. I'm not sure we've quite gone totally the full circle yet in any of them, but hopefully give you a bit of an idea of what we've been working on. So one thing I would say, however, is that over the past sort of 20 years, which begins to reveal my age when I say that's about how long I've been working in developmental biology. But over the past 20 or so years, I think there's been a huge shift in developmental biology. So when I first started, it was really a sort of very much a qualitative discipline. Experiments are incredibly difficult, they remain so. And typically we were essentially able to collect sort of qualitative observations and n numbers, so in terms of replicates, were incredibly low. But I think really in the past 5 years, we've seen a huge shift in developmental biology in terms of the research techniques and that we have available to us. And that really means that now the field is sort of becoming very much a quantitative discipline, so it's quite different. And I think that's really thrown up some challenges for mathematical modelers in terms of sort of how to make sense of all these data and how to really fully exploit the fact that we've got numbers, we've got quantitative data rather than just qualitative data. So we've begun to think, I think, much harder about how to do this. And I think that really brings up questions sort of pertaining to the fact that is, for example, my model at the right level for the data? Now, I think that was always there in modeling and qualitative data, but now I feel it's sort of slightly more at the forefront. And I might interpret that or turn that around and say, are the parameters of the model identifiable given the experimental data? Essentially, because I need them to be identifiable so that I can estimate them with any degree of confidence, and then I can quantify also uncertainty in those estimates. Because if I can do all that, I can ask questions about how uncertainty in the estimates of the parameters that I have propagate into uncertainty in model predictions, and therefore have so much of a sort of deeper understanding of the range of behaviours the model might predict, and therefore how that relates to the biology. So we're really thinking about this kind of approach. And what I want to do today really is to just give you a couple of examples of projects that we've worked on in the past 2 or 3 years, where we thought quite hard about this and really come down on the side of an incredibly simple model. But the fact that it was simple meant that we could calibrate it to the data and we could gain new insights. And it doesn't mean the projects are finished. I still think in both of the cases there are some more complicated models to be developed, but this was definitely simple was the right place to start. So yeah, I've said all that. Okay, so we're mostly going to be thinking about working in a Bayesian setting. So I just wanted to put that all up at the front. So given some quantitative data, can I conduct parameter inference using a Bayesian framework? So we're really interested for a given model in getting estimates of the posterior distribution, of parameters theta given data d. And we're going to use Bayes' rule to write that as being proportional to the product of the likelihood and the prior distribution. So I'm sure that most people are familiar with that, just wanted to get that out there. And I would say that in some cases we know something about the parameters a priori, but certainly in the second example I'm going to show you, we knew almost nothing. So it was interesting to see how far we could progress there. Okay, so into the project. So this is essentially a talk of two halves, two separate projects, sort of illustrating very, very similar points. So the first project looks at mechanisms of mRNA localization and essentially asks questions about what makes that process robust. So what is mRNA localization? So in very simple terms, it's a process that's used by lots of cell types, in particular larger cell types, to target proteins to the right place in the cell at the right time. And the idea is that instead of transcribing the mRNAs and translating the proteins and then moving those proteins to where they need to be, instead what the cell might consider doing is essentially putting the mRNAs in the right place at the right time and then letting them translate the protein when they get there. And it's been shown to be key in establishing the body axis, in cell migration, synaptic plasticity, etc. So what we wanted to understand is kind of what controls this process and what makes sure that it's robust. So if you're going to sort of prefer this process over one where you sort of target the protein sort of directly, then why should you do it? And how can you make it robust? And we are going to focus in the context of Drosophila because it's almost always an easier place to collect data than any other. So we're going to look at the regulation of mRNA localization in Drosophila egg chambers. So in this context, and I'll show you a bit more in a minute, essentially gradients of mRNAs are essentially crucial to set up polarity and establish the body axes. So this is essentially a Drosophila ovary, and it's got about 18 of these sort of strings that you can see going from sort of top to bottom in the image. And they're strings of maturing follicles called ovarioles. And essentially, they're sort of like an egg production line, right? So if you look right at the top, these are the most immature ones. And as you move down these strings, they kind of gradually maturing until they bud off from this sort of germarium. And what we're going to do is we're going to focus on a particular stage. So along one of these strings, we're going to focus on an egg chamber that has 16 cells. So a bit of a simple schematic to show you what the egg chamber looks like at that point. So it's got 16 cells, and essentially one of them is called the oocyte, and that's the one that's actually going to form the Drosophila proper. And then it's got a network of 15 nerve cells that essentially support its development. And what's important is that as this egg chamber has gone from 1 through 16 cells, via a series of cell divisions, that each one of those cell divisions has actually been sort of incomplete. So in the sense that after each cell division, the daughter cells are connected by a small intracellular bridge that we call a ring canal. So essentially, when you get this system of 16 cells that forms the egg chamber, they're all connected to each other, and they're connected in such a way that sort of intracellular components of the cell can be transported between cells. So in this context, we can see the movement of mRNAs between the nerve cells and the oocyte. And should say in this diagram, so everything here, it's wired in a very stereotypical way, right? This configuration is kind of essentially almost always maintained. And this cell over here that I'm just indicating would be the one that was the oocyte. Okay. So what else did I want to say about this side? I think just to illustrate the fact that a number of RNAs localize within the oocyte. So for example, gherkin, oscar, and bicoid, and we're going to be interested in the dynamics of gherkin in this work. Okay, so this is what an actual image, so taking sort of across a Z plane would look like in one of these egg chambers. So you can see the oocyte over here on the right, and the red sort of dots there, gherkin RNA that's localized in the oocyte. And then you can see 3 of the nerve cells at this level, sort of as support. And if I show you, just kind of go through this stack, you should be able to see all the different nerve cells eventually, and the oocyte. And as it plays again, I'm just going to point out a few things. So the oocyte was this one over here on the right. It's a bit slow now. Okay, it's about to go again. All right, so we've got the oocyte on the right. We can see the nerve cells and the red punctate spots, but we can also see these ring canals that pop up, and these are these connections between cells. So what's really great about having a sort of a Z stack like this is you can literally go through that stack. You can count the number of bright spots of gherkin mRNA, and that gives you quantitative data about distributions in each of the nerve cells and also in the oocyte itself. And Jonathan, my student who did all this work, was incredibly competent at being able to recognize from the Z stack like this which cell was which, and so to be able to to put numbers on it, which is great. Okay, so what do we know about this process? So we know a bit, obviously much more than this, but the kind of very simple story of what we know about mRNA localization is that these mRNAs are essentially only produced in the maternal nerve cells, so they're not actually produced in the oocyte, and they're transported to the oocyte through the ring canals. So essentially what happens is that multiple mRNAs are packaged up with some proteins into an mRNA protein complex, and then they're transported on the microtubule network of the cell by molecular motors into the oocyte, and that's what drives localization. So we came up with a huge number of different potential models that we could write down for this system, and we scratched our head for a long time and thought about whether we could calibrate them to data, and in the end this is the one we came up with, and it's incredibly simple. So we decided to build a compartment-based model where essentially all we're going to do is we're going to have 16 different compartments, one for each of the nerve cells and the oocytes, and we're going to simply track the number of mRNA complexes in each one of these cells over time. And then, so this is me showing you that same diagram as before, where cell number 1, which often is red, is the oocyte. It's always labeled as number 1. And then there's this very sort of well-established labeling pattern where 2, 3, 5, and 9 are always the cells that are linked to the oocyte directly by a ring canal, and then you get the kind of other cells sort of in this indicated pattern. So we've got biotransport of complexes between cells that are connected by a ring canal, and production in each nerve cell. So that gives rise to a system of 16 ODEs. Super simple. So you've got production at rate A, and this vector V just indicates the fact that you only get production outside the oocyte. You've got transport at rate B, and then this matrix capital B, which is a function of nu, encodes 2 things. So number one, the structure of B encodes the fact that not all cells are connected by a ring canal. So the IJ entry of B is only non-zero if essentially there's a ring canal connecting to those cells I and J. And then nu indicates the fact that transport is biased. Now, it's convention in the field to have nu representing bias away from the oocyte, not towards it, which is sort of back to front in my thinking, but I just want to point that out because otherwise some of the figures are going to look a bit strange later on. Okay, so that's a very, very simple model. And what's nice about that is you can write down the solution directly. And so when it comes to doing inference, everything is relatively as simple as it can be, at least from the model perspective. So you pick the right parameters and you get a kind of profile of mRNA across, or complexes across these cells that you might expect. So everything's localizing nicely in the oocytes. You see a lot of mRNA in cell number 2, and that's consistent with the fact that essentially cell number 2 has got a huge number of other cells kind of all feeding into it before passing on its contacts to the oocyte, etc. So this all looked fairly kosher. So we started thinking about inference, as I said, in a Bayesian framework, using a measurement model as indicated, so essentially negative binomial, which introduces 2 new parameters. So we've got sigma, which is related to the negative binomial distribution, but also another parameter which is hidden down here called phi. And this is essentially a kind of biologically required parameter, if you like, and it accounts for the fact that as, see if I can go back up, as these mRNA protein complexes are transported from these 4 cells that surround the oocyte into the oocyte itself, there's also some kind of higher order assembly process. So multiple of the complexes that you might see away from the oocyte are assembled together into sort of one bigger complex that's then transported into the oocyte. So this parameter phi is needed to be able to sort of take care of that process. Okay, so at this point the model is about as simple as it could be, and we've got quantitative data, and we began to think about how to parameterize the model. And the first thing that we noticed is that by having to incorporate this parameter phi, which is necessary from a biological perspective, you have identifiability issues in the model. So you can't sort of, from the data we have, cleanly estimate each one of the model parameters. So the idea that we had was to essentially do something very simple, which is to try and get a handle on that parameter, that assembly parameter first. So what we did was we looked within the nurse cells, within the oocyte and at the background, and looked essentially at the intensity of each one of these. So they're now purple spots that are the gherkin mRNA protein complexes. And we just analyze the intensity of each one of those spots, look at the distribution to get a handle on that parameter. So this is just looking at normalized intensity of those bright spots across backgrounds, across the nurse cells and the oocyte. And that gave an estimate of 0.4 for phi, telling you that on average about 2.5 of these mRNA complexes are stitched together, going from the nurse cells surrounding the oocyte into the oocyte. So the useful thing about that is that then you can use this estimate to specify a strong phi for phi in the full model, and then your identifiability issues are more or less sort of taken care of or accounted for. The other thing you can do then is to split so we've got a number of 4 parameters I think still to estimate, is that you can sort of split the problem up into 2 distinct parts. So the first is that you can look at the quasi-steady distribution of mRNA complexes. So essentially just recognizing that kind of at long times, the distribution across the nurse cells and the oocyte is really just driven by this parameter, this vector K2. So if you normalize all the counts at any time, that will give you essentially access to estimation of this parameter K2. And essentially what that does is allow you to estimate the bias parameter nu. So you can pick that parameter as well out sort of by looking at this quasi-steady distribution. And here's the results. So the prior distribution is in sort of darker purple and the posterior is in lighter purple. And what it tells you is essentially that transport is very strongly biased towards the oocyte. And just remember that in the experimental community, it's very standard to have nu being biased away from the oocyte. So the fact that this parameter is very small tells you that everything is on average being transported towards the oocyte and away from the nurse cells. And then you can do a posterior predictive check, just to so sampling repeatedly from the posterior distribution, and then simulating the model and checking essentially that your data lies within the range, but sort of predicted by the model. And things look pretty good, I think, there. And then finally, you can go back to the full dynamic regime and try and estimate the rate parameters A and B. So they were the production and transport rates. And I'm showing you the marginal posteriors here. So just indicating really that you can get a relatively good handle on both of those parameters. And that actually then those estimates of those parameters tell you that production and transport are carefully biased. So if I look at the production rate A, it's of a similar order of magnitude to B times the average number of mRNA complexes in any of these nurse cells. So things are fairly balanced. It's not one or the other that's really sort of rate limiting this process. Okay. And the other thing, again, I should say is you can do your posterior predictive check, sample from the posterior, simulate the model, and look at the range of outputs and how consistent they are with the data. And again, things I think look pretty good. So I've arranged this because it's going to be important in a minute in terms of sort of distance. So from right to left, this is distance from the oocyte. So on the right-hand side, you've got dynamics within the oocyte itself. Then the next column are all the cells that are 1 removed, 2, 3, 5, and 9. And then the next column, all the cells that are 2 removed from the oocyte, 3, and then 4 removed from the oocyte. Okay, and you can see that things look relatively good across the whole range. So super and fab, that's really, really good. But I always said that you kind of learn the most in biology when you're trying to do modeling when there's something you get wrong. So we tried to see to what extent we can sort of push our model to predict some behavior in different contexts. And one of those contexts was to think about an overexpression mutant, where effectively what you do is you try and sort of build a mutant where you have 2 copies of the gherkin gene in every cell. So by rights, a sort of very simple model extension would be to say, well, I've got 2 copies of the gherkin gene, so I'm essentially just firing out gherkin at twice the rate. And all I need to do to my previous model is stick a factor of 2 outside the production rate, and hopefully that means that I can then predict the dynamics in the system. So we went away and we did that, and here's the predictions. And sort of like at first glance, it doesn't look too bad. In particular, if you look at sort of the cells that are either the oocyte or the ones that are very close to it, the predictions don't look too bad at all. But then you start to scrutinize it a little bit and you realize that for those cells that are much more distant, they're on the periphery, so sort of 10, 14, 16, 12, things really don't look so good. So the model is in no way really predicting the levels of mRNA. They're much higher than we would sort of think. Then you can also look at what's happening in the oocyte itself a bit more closely. And so blue dots are from the wild type and reddish dots are from the overexpression mutant. So for me, sticking a factor of 2 outside the production term really just suggests to me that essentially I'd expect to see mRNA accumulating in the oocyte at twice the rate, essentially, in this overexpression mutant. But this plot here really says there's actual data. It really tells you that that's not the case. So actually, I've got about the same amount, or I'm seeing the same rate of localization of RNA in the oocyte. So somehow the egg chamber's buffering against this perturbation to essentially control the amount of mRNA in the oocyte. So the model in that sense is a bit of a letdown. It's not doing a great job in predicting the dynamics of this overexpression mutant. And that meant there's something that we'd not got quite right about the production or the transport process for this overexpression mutant. So we started to think about what we'd missed and what other mechanisms could be at play that would give robustness to the system. So we thought about blocking of ring canals. So is it essentially like a crowd trying to leave a stadium through very narrow sort of doors or passageways, and literally there's just too much mRNA in there to be able to be transported quickly through these ring canals in order to localize at the double the rate? Possibly. Could it be related to that we've got some density dependent transport process where essentially we're really, by putting 2 copies of the gherkin gene in there, you're really flooding the transport network and it can't cope. And so it's sort of essentially at maximum capacity already. And the other thing is maybe there's some inhomogeneous production, and this is probably the case. So when you do generate these overexpression mutants, they're not 100% valid. So we probably don't have quite 2 copies of the gherkin gene in every one of our cells. Okay, so there are some potential other mechanisms that could be at play. Can we think about incorporating those into the model and seeing which one of them or which combination of them might best explain or be consistent with the data that we have? So what we did, and I'm going to skip over the details here for the sake of time, was to represent these 3 different effects as a collection of models, so all combinations of these different effects, and then use some model comparison approaches to evaluate their plausibility. So essentially look at their ability to predict unseen data. And so here's the results. So model 0 is essentially the base model where we just stuck a 2 in front of the production rate. We know it didn't work all that well. Models 1, 2, and 3 are kind of each one of those different potential mechanisms that I described on their own. 4, 5, and 6 are a combination of 2 of them, and 7 is putting them all in. And so here's just very quickly the results of essentially this sort of model comparison approach and under two different ways of weighting essentially the model. So under the BMA weightings, we can see that there's overwhelming really support for model 1, and model 1 is the model that assumes that we've got some blocking of ring canals. So this idea that we're there's sort of a lot of crowding around the ring canals is essentially blocking passage of the mRNA complexes. And then if we look under slightly more conservative stacking weights, we see that actually model 1 is still the one that's most favoured, but also 3 and 5 are in there as well, probably with 5 being the next most consistent. And if I go back and look at 5 in particular, then one of the things that's got in there, and I said that this is probably true already, was this so it's got blocking in there as a mechanism, but also suggests that some inhomogeneity in the production rate. So the fact that we probably don't have 2 copies of the gherkin gene in every cell is possibly having an impact. So really the evidence from this very simple comparing, if you like, the very simple model and the data suggests that blocking of ring canals is essentially one of the biggest factors that's giving the system robustness in terms of the fact that it's not over, if you like, localizing mRNA in the oocyte itself. And there are more experiments that could be done here, but just by looking at these images that we have already, what we could see is if we looked in the region of a ring canal, we could really see this buildup of these mRNA complexes supporting that hypothesis. So I'm halfway through the talk, and hopefully I've got about half my time left, but just very quickly, the conclusions I would draw here are just that if we have a simple model, but we can connect it to data via Bayesian inference, then it can be really powerful in distinguishing between hypotheses. So in this context, for gherkin mRNA localization, we think that there's a tightly regulated balance between production and transport, and that crowding of these RNA protein complexes at the ring canals helps to regulate the process and make it robust. So certainly that there's much more that can be done here, but I think at a very high level that was they were sort of nice conclusions to be able to draw. And I think going forward, what we need to do is interrogate those processes sort of a little bit more deeply, maybe using some different types of data, but also some slightly more complicated models. At the start, it was pretty good. Okay, so I'm going to take a total about turn now and tell you about some work we've been doing a lot more recently that tries to sort of take the same approach in terms of building a model and calibrating it to data to draw some mechanistic conclusions. But it's a different level, and it's all about thinking about how electric fields impact single cell motility. So I said that in the Greek we've been thinking about for a long time about collective cell migration and the mechanisms that could drive that. And so really interested for a long time as well in thinking about directed cell migration. And we know that the cells, or many cell types, have the ability to respond to a huge variety of environmental cues. So maybe one of the most well known is chemotaxis, as illustrated in a video on the right-hand side, where essentially we have a diffusible chemical that's emanating from some localized source. And essentially, as that chemical diffuses and potentially decays, then that sets up a chemical gradient, and then cells essentially want to move up gradients in the chemical, and that's known as chemotaxis. But we also have other mechanisms, for example, haptotaxis. So that would be movement up gradients in adhesion sites or in substrate-bound chemicals, and durotaxis, where this is essentially movement up or down gradients in, for example, matrix stiffness. But one of the sort of cues that cells can respond to that isn't so often talked about is electrotaxis. And this is directed motion of cells due to an applied electric field. So a huge variety of cell types can naturally sense and actually respond to DC electric fields. And maybe that's not surprising, because actually such fields arise very naturally, both during development, but also in wound healing. So you might expect it. And what I really think is interesting about electrotaxis is that it's got huge controllability. So compared to some of these other cues, it's relatively easy to go into an assay and use a system of electrodes to control a population of cells. So what you're seeing here is on the left a population of MDCK cells, where the field is left right for a bit of time and then up down, and you can see they really undergo this sort of quite sharp turn in their metabolism in terms of their direction of motion. And it's in the cell population over here on the right, it's a little bit less pronounced, but you can see that for the first, and it's not marked, but for the first portion, you can see them moving up, and then partway through the experiment, the field is reversed and they all begin to move downwards. So we can see that the cell populations respond very nicely to electric fields. And there's been already some thought to using sort of this concept of electric field guided cell migration to develop new therapeutic options. So here I'm just showing you a set of experiments that were done to try to showcase the potential for bioelectric plasters. So the idea is that you would, within your plaster, try to establish one of these electric fields so that the field would encourage the cell population to move in the right direction and to close the wound. I'm not sure it's there yet, but what I really find is fascinating about this is that the potential for, I think, modelling to make a contribution to our understanding of these types of processes, partly because it's just so controllable. So our real long-term aim is to think about developing electrotaxis into some kind of cellular bioengineering tool that can be used in the context in which I just showed you. But I think we've got a long way to go yet. We're right at the start of this. So we're going to think about single cell behaviours, and then down the line, and we just started thinking about that, is how to scale this up to populations. But I want to tell you today about is all the stuff we've thought about for single cell behaviours. So we know that in the absence of electric field, there are a huge range of models out there to describe the motile behaviour of single cells. And we've learned a lot from a number of research groups about how to scale that up to think about collective migration, again, in the presence, or sorry, in the absence of an electric field. So we're sort of taking the view that the thing to do next is to think about what happens to single cell motility in the presence of an applied electric field, and then eventually be able to put that all together to think about the motion of cellular collectives under those fields too. So that's our sort of goal, sticking at the single cell level. And I should say that we're going to stick at a relatively high level when it comes to modelling the motile behaviour of these cells under applied electric fields, because there's simply so much we don't understand about what happens sort of inside the cell in order to be able to transduce the electric field signal and turn it into movement. And just to give her an example, if you took a different cell type that you'd never tested before, there would be a priori no way of understanding whether it was actually going to move towards the anode or the cathode, and different cell types move in different directions in the same field, which I find pretty interesting and exciting, and hopefully someday we can get to understanding that. But for now, we're going to stay at a much higher level. Okay, so these are the data we're working with. We've got control data where we've got human corneal epithelial cells imaged over a 5-hour time window at 5-minute intervals. And so we can essentially, we're going to assume that the cells, and this is an assumption, that cells aren't really interacting with one another. So we can consider single cell behaviours. And we are going to collect tracks from each of these movies, and about on the order of about 25 or 26 of them. That's going to be our sort of control data. And then we also have data with an applied electric field. So for hour number 1 of these movies, there's no field. Then for hours 2 to 3, so this is hour 1, and then 2 to 3, you can see that the field is from left to right, so they all try to move towards the right. And at the beginning of hour 4, that field is reversed from right to left, and you can see the cells move in the other direction. So again, assuming that this is sort of single cell motility, we're going to collect tracks from about 25 cells from each of the experiments and use that as our data. And I'll try and remember to say it again, but actually in the calibration process, we're going to keep the data from hours 4 to 5 where the field is switched. We're going to keep that back as data with which to sort of test the model. Okay, so I'm just going to walk through the steps of the model, how we thought about developing it, and then how we calibrated it. So it's a pretty simple model, an SDE model where position of cell X changes because it's got velocity V. And in the absence of an applied electric field, essentially the velocity is all driven by what's happening with the cell polarity P. So we've got a parameter V, which is essentially the modal cell speed, but it's all down to thinking about how to model changes in cell polarity over time. So the model for the evolution of cell polarity, so some drift diffusion SDE, where essentially we've got a parameter D, which is essentially like a diffusion or a rate parameter, and then we need to establish a sensible form for the potential function W. So we didn't want to go too deep into this and go too complicated with this. So essentially the thing that we did was to eyeball the data from the control experiments, just looking at cells when they were moving autonomously. And so if you look at this distribution, these distributions of displacement distances, so each one of these lines is a different cell, you can see that a kind of a consistent modeling assumption would be the fact that cells would spontaneously polarize, in which case they're going to travel at some positive speed, and so they can also transiently depolarize. And you can see this if you eyeball the movies as well. And then looking at the autocorrelation in terms of their direction of movement shows you that sort of drops off with increasing time lag. So again, consistent with a kind of drift diffusion sort of or random walk type model for autonomous cell motility. So given all that, we tried to come up with an incredibly simple model of essentially for the cell polarity. So what we know is that cells spontaneously polarize, and once they do polarize, in the absence of a field, their direction of motion is in bias. So we can write W as a function of the modulus of P, so just sort of non-vector P over here. And then we chose this form for P, which basically just tells you that essentially when they're polarized, that P is going to, the polarity is going to stochastically evolve in a regime where the modulus of P is approximately 1. And then when I put the full model back up here, then you'll see that then that just gives you that cells move essentially at a speed V. Okay, so that is what the model looks like. And so what have we got within this model? It's quite simple. It's just got 3 parameters. It's got this timescale parameter D and the parameter beta of the potential function, which can also be thought of in relation to a potential energy barrier delta W. So that's what I'm going to work with. And those parameters control essentially the rate at which cells transiently depolarize. And the other parameter that you've got in there is the parameter that relates to the cell speed. So 3 parameters to estimate from the data. Okay. So again, taking a Bayesian approach to model calibration, and first of all, we're essentially trying to access the posterior distribution of model parameters theta, given data that's collected in the absence of an electric field. And to remind you, we've just got 3 parameters to pin down. So in contrast to the other case, the likelihood for this model is intractable. So we had a number of options, and we decided to go with using synthetic Bayes to estimate the model parameters or the posterior distributions. So essentially here, what we need to do is to define some summary statistics of these trajectories. I can show you what they are if you're interested. And then essentially what we're going to do is approximate the likelihood with the model with the synthetic likelihood. And essentially that's the likelihood of this summarized data. So looking at using the summary statistics under the assumption that they're Gaussian distributed. So if you do that, gone backwards instead of forwards. If you do that, then that leads you to being able to generate some posterior distributions for your model parameters. So on the diagonal, as ever, I'm showing the marginal posterior distributions for each one of the parameters. So the cell speed, the depolarization barrier, and the timescale constant. In the lower sort of triangular part of the plot, I'm basically just showing you the bivariate posteriors. And really, the sort of reason for showing these was to illustrate the fact that we had almost no idea what values some of these parameters should take. So we took quite wide prior distributions, but that the posterior is really localized to quite small regions over that prior space. And in the top sort of triangle, you can see much more zoomed in sort of plots of those posterior distributions and the bivariate ones. Okay, so it all looks relatively good. It looks like we can get a fairly sensible handle on most of the parameters or on the parameters of the model. And you can do some posterior predictive checks, again, sampling from these posterior distributions, simulating the model to check that you've got agreement between the model and the data. And because we were doing synthetic Bayes, we looked at the level of agreement between the summary statistics, so there's the 4 of them down here, generated by the model and coming from the data, and I think things look relatively good. And we also just sort of double checked that as we looked at the 2 different replicates and we separated them out, that things look relatively similar between the 2 replicates, and also if we looked at the kind of combined posterior, things didn't change too much either. And then you can also go where you don't have to stay at the level of summary statistics, but you can look at, so this is a number, or the trajectories that we have from the control data all sort of pinned to start at the origin, and you can look at kind of how those trajectories compare with those that you might simulate from the model. I think they probably look, using the eyeball norm, reasonably good. And you can also use then the model to make some predictions of cell characteristics that it isn't necessarily easy to estimate directly from the data that you're collecting. So for example, what's the probability at any time that a cell is polarized, or how long does it take for a cell to polarize or to depolarize? So there's some nice things that you can essentially get from the model that you can't easily estimate from the data, but you can have a stab at estimating because you have calibrated the model to data, I should say. Okay, so keeping on going, because the whole point of this was to think about what happens when you apply an electric field. We started to think about how to build in the impact of the electric field into this model. So the field that we've got is 200 millivolts per millimeter, which has got a single field strength. But just to remind you, basically, the direction of the field is flipped partway through the experiment. So how does that impact cell motility? So the idea was to essentially take the model that we had for autonomous cell motility and to think about all the different ways, much like we did in the previous project, where motility could be impacted by the electric field. And we came up with 4 that made the cut. So the first is just a velocity bias, which I think can be interpreted as saying that the electric field imparts an additional force on the cell that's in the direction of the field itself, and it has magnitude gamma 1 relative to essentially the force coming from the polarity. The second way that we thought the field could impact motility was just to increase the speed of the cells. So essentially, says don't care what direction the field is in, it's just the sort of magnitude of the field that's important in increasing the cell speeds by a factor gamma 2. The next one was speed alignment, and what that says here is that cells are going to increase their speed or change their speed in the presence of this electric field, but the extent to which it's increased or decreased is comes from how aligned the cell polarity is with the field itself. So p hat, sorry, that's a bit of a strange place, is a unit vector in the direction of polarity, and then you just take the dot product with the electric field. Just telling you that if the polarity happens to be aligned with the electric field, then there's a speed increase. If it's totally in the opposite direction to the electric field, there'll be a speed decrease, and everything in between as the angle just varies. And then finally, we thought about putting the effect of the field in a different place in the model, and that's in the equation that controls the evolution of polarity. And really what we said is that here we've got the assumption that in the presence of a field, the cell polarity is going to be preferentially biased in the direction of the field. Okay, so 4 different ways in which we can impact cell motility. 3 of them are really easy to just showcase using a plot of the steady velocity distribution. So under the autonomous model, a cell wants, essentially when polarized, will have speed approximately v, so that's the radius of this ring you can see here, situated at the origin, and everything's nice and symmetric because the cells aren't preferentially polarizing in any particular direction. So essentially, most of the time they go and undergo a random walk sort of around this ring. And if you switch on the electric field, these are how 3 of the 4 potential mechanisms impact the velocity distribution. So the speed increase parameter gamma 2 essentially just increases the radius of that ring. And then gamma 1, which is this sort of the fact or the assumption you have an additional force on the cell in the direction of the field, just translates this ring to the right by a distance gamma 1 v. And then gamma 3, which is essentially relating to how aligned the cell polarity is with the field, just essentially shifts this ring from being symmetric. So it puts much more weight on the right-hand side because that's where polarity is aligned with the field. So that's a kind of, I think, provides a nice way of looking at the different impacts. Okay. But let's go back to calibration of the model to the data. So we've got now 7 potential model parameters. So things got a bit harder, and we again want to do inference using a Bayesian approach using synthetic Bayes and synthetic likelihoods. But this time we've got data both from in the context of having no electric field and data in the context of having a field. So we can look at those 2 likelihoods sort of separately. And just to remind you that we've reserved the portion of the data where the electric field direction is reversed for sort of testing the model later. Okay, and this is just a reminder of what the data looks like. Okay, so what was the idea? We've got 4 different possible electrotactic effects. So that could give rise to a collection of 16 different possible models. So this is all combinations of these different potential electrotactic effects. So model X is going to be basically a model that's got some subset of the effects 1, 2, and 3, and 4 from it. And then you can do many things for model comparison, but we defined an objective function, which basically just looks at the support for essentially the data under model X. And the main thing to note here is that each one of these models has slightly different prior distributions just because it's got different numbers of parameters in it. So if you look at model comparison, in terms of this objective function, and sorry, there shouldn't be a mu there actually, but we can just set mu to be 0. These are the results that you get. So essentially a higher value of J indicates more support for the data under a given model. So you can see that essentially for a model that just includes effect number 4, that's the one that's essentially best at predicting the data that we see. Okay, and then as you sort of go down the list, any of the next models that you see all have this impact 4 in it. So just to remind you, essentially this this model 4, if you like, or effect 4, was the effect that cells essentially are going to preferentially polarize in the direction of the electric field, and that so really essentially what we're going for here is saying that what we think is happening is that cell motility, or this sort of biased cell motility, arises solely from the fact that cells are going to preferentially polarize in the direction of the field. So it gives you a model that looks something like this, where essentially all the effects come in through changes in how the polarity evolves over time. Okay, probably should go back then and look at the posterior distributions. Again, they all look pretty good. We can estimate all the parameters relatively confidently, and we can do our posterior predictive checks in the context of having the electric field around there. And again, the comparison between the summary statistics predicted by the model and the data, I think, look pretty good. And you can also simulate essentially from the model, both in the context of having no electric field, which is what's going on in the top row here, and in the presence of an electric field. And you can see that, I guess at least by the eyeball norm, these trajectories don't look too dissimilar to one another. So all was looking relatively good here, but the thing that we wanted then to go away and do was to test how our model now fares on the data that we held back. So this was data where we essentially reversed the direction of the electric field after 180 minutes. So we can use our model to make some predictions there. So if you just look at the level of the summary statistics, you can see I think there's a relatively good agreement between the model and the data. And so I don't think we're doing a bad job, right? So cells are certainly essentially reversing their direction of motion as the field is switched in direction. So they now want to move from right to left. But I would say that at the level of looking at the trajectories, you begin to see some slightly more obvious differences between the predictions of the model compared to the data. So in the observed data, so this is data from the experiments, you see that cells very, very rapidly turn around and start moving in the opposite direction when the field is switched. Whereas when you look at the predictions that come from the model, you can see quite a number of cells who kind of really take their time to essentially sort of change direction. So it sort of suggests that in some senses the polarity isn't quite changing quick enough to replicate the dynamics observed in the experiments. So pretty happy with these results, but I think that's something that we can do to maybe improve things going forward. But why do I think they're not quite 100% here? I think there's two things that actually could be going on that actually is more kind of model or process driven in terms of our analysis than really kind of the fact that we've got the biology wrong. And I guess one of them comes down to the fact that we really, if I could just stick it back up, sorry, we really assumed that we were looking at single cell data, whereas we can see that there are interactions between cells, and potentially at the level, you can see it there where the field switches direction, right? There's bunches of cells that do interact with one another, and maybe basically the data that we're getting is essentially, it shouldn't be modeled as if cells were sort of single cells that aren't talking to one another just a little bit. So that could be a reason for some of the discrepancy, and maybe what we really just need is data at lower density. But the other thing could be the fact that we've used synthetic Bayes, and as a result of that, necessarily we've had to use summary statistics of the cell motion in order to make progress in estimating the posterior distributions, and possibly those summary statistics aren't quite capturing the behaviour that we're seeing. So some things to think about for the future, I think, for this project, but overall relatively happy and to draw the conclusion that the observed bias in cell motility is really coming from cells wanting to polarize in the direction of the field as opposed to anything else. What I think is nice about this work, and I'm quite excited about, is being able to apply this framework to other experimental scenarios and cell types. So I'd like to see if we could vary the strength of the field and repeat these experiments, which is easy enough to do. Would we draw the same conclusion? Would it always be kind of a mechanism 4 that wins out there? And can we get a handle on how this parameter gamma 4, which controls the sort of strength of the effect, can we get a handle on that, how that relates to the strength of the field? And then in the future, I think I hinted at this at the start, we're really at the moment starting to think about how to extend this model to populations of cells. And my sort of dream scenario is to use the model to develop optimal stimulation protocols kind of on the fly. So we're kind of simultaneously imaging a cell population and then thinking about how to adjust stimulation protocols to make them move in a particular direction or in a particular way. So to just summarize, what I've tried to highlight is that because we've got so much now quantitative data in sort of cell and developmental biology, we should really be thinking about calibrating the models to data. And I think I've shown that using even if you're using sort of very, very simple models, you can still draw conclusions when you combine them with data as to what sort of mechanisms are at play in a given system. But hidden under all the hood of this was the fact that determining the right model is often really, really difficult and is definitely data dependent. And therefore sort of analysing model identifiability can be really helpful during the model building process. So I took out a huge portion of this talk to make way for the Electrotaxis stuff, but there was a lot in there about identifiability, and I think that is certainly the way to go. And hopefully showing that a Bayesian approach enables us to quantify uncertainty in the estimates and in the model predictions. So with that, I'd like to just say thank you for listening. It's 2 parts of this work were done by sort of 2 different groups of people, really. So the Electrotaxis work was all done by Tom Prescott when he was a postdoc in my group. He's literally just started at Turing Institute. And that was in collaboration with Minjal's group at UC Davis. And Kanju was the person who did all the experiments and tracked all the cells for us. And then the mRNA localization project was done with Jonathan Harrison when he was a PhD student at my group. He's now at Warwick. And that was in collaboration with Elan Davis's group in biochemistry in Oxford, and Richard Parton was the person who did most of those experiments. So that's me, and just like to thank you for your attention, and happy to take any questions.